Yeah, my name is Simon Geist. I'm an assistant professor for marine fisheries at the Texas A&M University Corpus Christi. Came over here in 2015 from Bremen, the ZMT, where I worked in the labs of Werner Ekau and Andreas Kunzmann on um, questions related to larval and juvenile fish in the Benguela current within the genus project, where I know Bettina from, and several others, of course, were on this meeting call. Come on. There we go. So larval fish and juvenile fish. So I would say, like, why should we study them? Of course, like Chris mentioned, it's like important to like understand the recruitment and the stock recruitment relationship. Like that's like also what Dave Cushing pointed out, one of the big names in that field. I would say if you are more into fisheries management, then like the typical type of studies that is done around the world is monitoring of like early life history stages, larval surveys here in the Gulf of Mexico. NOAA, like the federal agency of the US, is doing like a larval surveys several times of the year. And then also juvenile fish surveys are done by the state agencies, like by the Texas state agencies here. I know that at NUTMERC, I think you included like the larval fish into your regular environmental surveys, at least like to some point, Richard. You can correct me or update me later on that. So there's a couple of like crucial overall theories related to like recruitment success, and like several of them are like focusing on like different aspects of tropho and hydrodynamics, the critical period and the aberrant drift hypothesis of like Johan Jort's, like where it all began in 1914, 1920. And then there's others like the one of Cushing is like focusing on like the availability of like food and the mismatch of like food and the availability of like a or presence of larvae at a certain age as well. And of course, if we talk about like upwelling systems, Curry and Roy and like Alaska, also a stable retention hypothesis that focus more on the hydrodynamics, kind of like the optimal upwelling intensity and like what comes with it. Another important hypothesis or like theory that is like I would say stated to be generally valid for like a larval or like early life history stages, developed by Ed Hood in 1987 in his paper, is and then kind of like further developed until today, and like this is also something we are like testing, or many people who are like working on larval fish are testing in one way or another, is a stage duration hypothesis. This graphic shows that there's like predation, starvation, and of course also physical process and disease like act on like eggs, the yolk sac larvae, then the larval stages, and also the juveniles. The stage duration hypothesis assumes that like a faster growth is like beneficial for survival, because that will get you out of, as a larvae, that will get you out of like the larval stages where like kind of like movement is limited, movement ability is limited, and kind of also gets you out of like the prey window of like some predators, and like assuming that like as a juvenile, you are then more capable to escape some of these risks. Another important theory, and that's like more for the physiology, but you have to like understand the ecology and the physiology of like an organism together, of course, and that's like is like the thermal window theory put forward by Pertner, different co-authors over the last 2 decades, I would say. Specifically, there's like some critique, but I think it has like kind of like about like the specifics of the specific form of like these relationships, but like basically it understands that temperature is like the main driving factor that is like confining the presence and like performance of like of an organism. And then you have like other factors like CO2 concentrations, like ocean acidification, hypoxia, or also in the one for what is interesting for our situation here in Texas, like salinity, being like factors that can confine like the environmental window, meaning like kind of like the environmental settings in which like a species thrives, and like not only a species, it's like can be different for different life stages. In the research that I'm doing and I'm interested in, it's kind of like trying to like match these two approaches, like ecology, trying to understand like the ecology and like the physiology and how it like relates to the environment like early life stages encounter in their habitats. The next slides, I want to show you like some of like the study or like the research we did like in the Genus project. Bettina already introduced it to you. Once again, I was like working in the group that looked at the ichthyoplankton specifically in Werner's and Andreas' labs. And I just skipped this because like Bettina mentioned like the overall goal of the Genus project, which was like kind of looking at like how climate change, nutrients, and greenhouse gases and the structure of the ecosystems in the Namibian upwelling system are can be better explained, and kind of like interrelationships can be carved out with a focus on processes and mechanisms, not so much about like abundance. So like kind of like the other part of research, and that's like where usually university researchers and also myself come in, is not looking at like those monitoring programs that are like very often done by like federal or state agencies, but like trying to understand, like picking out like trying to understand processes that lead to like what you see in terms of abundance and presence. The Northern Benguela current, cold nutrient-rich water, everybody knows that in the audience, I guess. And we have like here like around Wallfish Bay, like an important, like historically important, like fish recruitment area, and then another one up here between the Northern and the Conini Sill, especially for the sardines, which was like important in former decades, like not so much anymore in recent years. The work we did in the Genus project was like threefold. So like one, we were interested in like the tolerance of hypoxia exposure. And we used like methods of respirometry, like measuring metabolic rates. And also we had like a master student, Rashid, who is now with the Kenyan Ministry of Fisheries, who looked at the activity of metabolic enzymes. That manuscript is kind of like difficult to get through, but there's like another take on that was taken earlier this year. Fingers crossed. Trophic ecology was like a big focus. I looked at stomach content of like horse mackerels, and then it was the main species. And the idea was to compare it with like sardine and anchovies. Problem with like those two was that like the way we sampled was like not a lot of stomach content in them. It's like just like caused by the morphology and the shape of like their stomach. It's easy to evacuate for them. So we also looked at like stable isotopes, fatty acid trophic markers, and once again combined that with like the respirometry, because metabolic rates can tell you something about the energy demand of like a specific organism too. And then in one of our last cruises for the Genus 2 program, we also had like a Steffi Simon with us, who did some starvation experiments for her master thesis. And combining this, of course, there's like a couple of integrating proxies. You could say like respirometry and metabolic rate is an integrating proxy, but there's also like otolith increments that can tell you something about like the growth rate of fish larvae, the morphometric condition factor, either in its simple form, the Fulton condition factor, just like comparing like weight at a certain size, we are like exploring in one of the projects here in the Gulf of Mexico. We are also exploring like a little bit more sophisticated one that takes like 6 morphometric measures. Then we looked at lipid content and also RNA DNA, all in relation to spatiotemporal distribution. For the Genus project, we did 7 cruises. We used like open and closing nets, like multinet with a CDD as a main gear, and also supplemented that with like catches by ring nets and a chukka troll just to collect like both like larger fish larvae and also just a larger amount. They were just like from the diameter, they were larger. I would say in general, I don't know if you did that in traffic. We had like from Bettina and Rolf and like the Hamburg colleagues. There's of course one other method that they had like an instrument available, but wasn't like deployed in our last Genus cruises, which is like a towed video camera system that allows you like even more specific kind of like depth specific kind of like fine scale relationships and like kind of the vertical distribution of like different organisms. With those multinets or like the mock nest, which is like a larger brother of it, you have like depth spins that are still kind of like several tens of meters wide in the vertical dimension. So it's a bit more difficult to explain like fine scale patterns of like overlapping of like gelatinous plankton, for example, and larval fish. So hypoxia effect on larval fish. This is from like a paper of ECO and like the Genus team. You see here that like there's like a shoaling of the oxygen minimum zone, the yellow, and then kind of like the collapse of like the sardines and the anchovies with like the blue, the horse mackerels like still doing pretty well, if I'm correct. And like so like from this observation, like the idea was like maybe like the hypoxia, the shoaling of the hypoxic zone is like one contributing factor to like the problems of especially the sardines to like recover. Looking at the vertical distribution of like fish larvae in relation to the oxycline, that is like the first thing that was done by Werner and in a paper, and like I think Holger was involved and Dr. Hagen and also Dr. Verheyen in the 2000s, and they found there was like kind of like a different vertical distributions based on like different species of fish larvae based on like those multinet catches. And we saw it again like in the samples that we took during Genus. Metabolic rates, and then the enzyme activities. Here like pictures, gobies, hakes, and we're like taken into account for like the enzyme study, but like the focus was on horse mackerel and then sardine and anchovy, like those long and slender guys here to the right. This is like the figure from the 2018 ECO et al. Paper, and you see that like the gobies from those multinet catches, they seem to extend like farthest and like, but also like in growlers, this is the range and this is like the median. So like Cephalogobus bibarbatus, the pelagic goby, Maluxius, more or less like in the same kind of like depths and like kind of like gobies, as it's known for like from Bronwyn and her colleagues and Norwegian colleagues that like they like to, or they can stand like to be like in hypoxic conditions longer. So like that was like shown here also for the larvae. Whereas like the sardines are like more up here in the water column and kind of like also leading us to the hypothesis that they are like not as well prepared for like hypoxic conditions. For the gobies from like a Norwegian study, we know that like they have like a very low critical oxygen level from respiration studies, like a 5.3% oxygen saturation, and they are it's like where they get like in trouble with their metabolism. From experiments we did on juvenile horse mackerels, we saw that they are like not as well adapted as the gobies, but like also still pretty good. 13% like was kind of like identified as a critical oxygen level here, and from the larval horse mackerels was like more challenging to like measure at that time. We have like a nicer, better equipment now here in my lab, so it'd be nice to repeat that one day. or like extended. So like what we can only say like from the experiments that we did back then, that like there was no significant decrease in respiration until 30% oxygen saturation, which is also like a pretty low oxygen concentration for like larval fish. Like many other species, they're like 60% or even over 60% is like the threshold where they get in trouble and show like signs of problems. For the enzyme activity, just like here, like quick on the right-hand side, like pyruvate kinase and lactate dehydrogenase are like metabolic enzymes that are related to like the anaerobic metabolism, and gobies were like had higher levels of Cs compared to the other species investigated, and like the sardines had like the lowest, like both as like a low activity level. So we think we would infer that like this is an indicator that gobies and also horse mackerels are better prepared. Of course, like you can argue that like those enzymes are also used like for other activities and like kind of like just like regular swimming activities. So it's not like a like that was one of the critiques in like the reviews, that it's like not a standalone indicator of like hypoxia tolerance. But and I agree with that, but like I think it's like still a hint. It would be interesting to like look further into that, like with other markers at a molecular and a biochemical level, eventually to like carve that out. And that is for larvae only. If they would be like better, if there's like species that are better prepared than others in terms of hypoxia tolerance, that could mean that under hypoxic shoaling of the hypoxic layer, like some like the sardines, if they're like less adapted to like withstand hypoxic conditions, they couldn't like go into like this retention cycle here, where they like migrate into like deeper waters, where they encounter countercurrent, like it brings them back to the shore into like the good feeding grounds over the shelf, and they are attracted to offshore waters that are like then not as conducive for like survival and kind of like recruitment, affecting recruitment. That's like our hypothesis, taking it from like a Norwegian modeling study and like putting into our hypoxia results. Let's see. Food web, like Bettina talked about like the food web in terms of like the gelatinous like plankton. We looked at like larval fish, specifically like those three species that I mentioned, sardine, anchovy, and like horse mackerel. There's like a theory from Carl van der Lingen et al. That there's like different food chains, right? Like depending on the upwelling activity, you have like either larger or like smaller plankton, and then kind of like favoring either or of the two pelagic fish species. So that was our starting point. We also know from like a copper time series that there was like a decline in total copper density during the 2000s, and like a higher importance of cyclopoida and especially ozona relative like to other groups in more recent years during the Genus project. And that is why I looked at like stomach content composition of like horse mackerels as the main species, and like we saw that ozona is like the important food source throughout the larval phase. So they can exploit like this relatively abundant, relatively small copepod compared to like the larger calanoida, but like when they're growing in their body size and metamorphosing into juveniles, there is like a diet shift towards the larger calanoids. Taking like the information from like this stomach content and like combining it with the metabolic rates, and also with like some carbon content data from my colleague Dr. Anna Schuckert from Dr. Hagen's lab. We were then able to like estimate like the minimum prey requirements in terms of like for like different sized horse mackerel larvae and juveniles at 5 sizes, estimating like the numbers of like different copepods that they have to ingest. And you see like that kind of like yeah, they have to feed on once they are 10 grams, and if they would only rely on like those small copepods, it's kind of like numbers get pretty high. So that's probably like unrealistic for them. So like pointing to like yeah, a problem if like there's not enough like larger prey items. Potential problem. We did like some, and this is like still not published, and like, not sure if the starvation part will ever publish, but like we have like some RNA field data that are finally working up this year and starting last year. But like we saw like in this kind of like pilot studies, there was a starvation effect like visible within like horse mackerel larvae, like after immediately in the RNA DNA signal, but like until even after 70 hours of starvation, it was still above the threshold of like that was established like with like the RNA DNA method like for other species. So it seems to be like just from this like small data set, it looked like that they are like can overcome like several days of starvation without a critical problem, causing a critical problem for the survival for the horse mackerels once again. So they are like pretty tough, and that's maybe like one of the explanations why the horse mackerels are doing better than the sardines in the Benguela. We also looked at like with like some statistical models. We try to like identify like the environmental windows, and we saw like that for only for like the small size class from 3 to 4 millimeters, there's like this interesting nice shaped hump indicating that the 18 to 20 degrees Celsius temperature at 20 meters seems to be like the sweet spot here for like the small horse mackerels. Not so much going on for like larger horse mackerels in terms of temperature and DO. So like meaning probably pretty, once again, like speaking to like the interpretation that they're pretty robust over like a quite a broad spectrum of temperatures and dissolved oxygen levels.
Simon, can I give you 1 or 2 minutes to wrap up?
Sure. Okay, so like this is like in like this genus data, like we saw like the horse mackerels are like in different aspects like doing better than sardines in terms of their larval stages and their biology and physiology. And like just like 2 minutes to go over like different projects that we do in my lab at the moment, or have been doing since I came over here. One big one is like a joint project for which we did like 3 cruises in with like colleagues from Louisiana and North Carolina, where we are looking at like the effect of Hurricane Harvey on planktonic communities, bringing like the genus to like the Gulf of Mexico, so to say. No, like Hurricane Harvey caused like the largest freshwater plume ever recorded, or like it was the largest rain event ever recorded, and caused like a large freshwater plume that went into the Gulf of Mexico and affected especially salinity and like shelf waters. So we are interested in like how that changed like the food web and also like growth condition and abundance of larval fish, and also like if it caused an expansion of like spawning areas of like coastal larval fish species. Then locally, we have like a small boat available where we do like ichthyoplankton studies around Corpus Christi Bay. Like Texas has like lots of like shallow bays that are have like a vast seagrass and oyster reef habitats, and like very important nursery grounds for like several species of fish, many cyanids. Like I think in Namibia and like South Africa, like it's like the dusky cob is like a cyanid, right? So like relatives to like the cob here. So we looked at like ichthyoplankton ingress through a small inlet, like a food web during that passage of like the inlet, and like a new project is looking that just started now. We will look a small project. We will look at the vertical distribution of larval fish in relation to like during like the passage of the inlet, because and that's maybe like a relation to Namibia as well. I don't know how much like concern is about like fresh water along the coast of Namibia and how many desal plants you have, but like here, there's plans, massive plans to build like desalination plants, and like also Corpus Christi is the largest exporter of like oil in the US, the port. So they are like also increasing like this export facilities, and like they're planning to build like desalination plants. And there's of course concerns like for what are the effects on like larval fish and larval shrimp. Microplastic pollution is another topic. We did like 2 projects, like the second wrapping up now. So like just like to look at a baseline of microplastic ingestion. In short, we find like microplastics as in many other places in many in like the majority of like the juvenile fish that we studied, and there seem to be like some differences between species, and like but like yeah, consistent patterns about like feeding gills is like difficult to extract, I would say. And the last one is like the ecophysiology of like hatchery-reared larvae for like the state agency stock enhancement program. That's like a more a physiology project with Parks and Wildlife. They are rearing southern spotted sea trout, which is cyanid, and then southern flounder. And we look at like temperature and salinity effects on metabolic rates. And we also like just start a project where we will release like flounders, juvenile flounders in 2 different sizes into the wild to see like how they into like net pens in the wild environment, to see how they do in terms of mortality and like some other parameters in the first 2 months after release. Okay, I think with that I wrap up. Sorry for taking a bit long, and open for any questions.